Our next presenter, Dr. Mark Rossenberg, is going to do a presentation that is truly near and dear to my personal life. Those of you who have attended my class, those of you who have watched me moderate, and those of you who have seen me lecture will see that I wear a band and I wear a cancer ribbon due to my own personal tragedies, which I won't share right now. Some of you have heard of it. Many of you have. Some of you have read my writings that I've written about. Cancer is very near and dear to me, and it's a combating demon and a devil, and sometimes we just don't know how to combat it. But it is a cause that I personally, wherever my life will take me, will choose to submit and to cherish and to honor for the rest of my natural life. Dr. Rossenberg studied the mechanisms of cancer treatment failure and following concepts that were blatantly apparent to him. And there's many substances that are toxic to the cancer cells in vitro. But what he tried to figure out that these therapies, however, are often more not prevalent in vivo inside the body. And the primary reason for the ineffectiveness of these tumors is proper blood flow. Improper blood flow can possibly increase the size of these tumors. And I won't steal his thunder, but I will share with you that I wish I had personally met Dr. Rossenberg a couple of years ago. And his fruitfulness and his education and his treatment may hopefully prevent some personal tragedies that I and millions of people go through each and every day. It's my pleasure to welcome Dr. Rossenberg.
Thank you very much. Thank you. Thank you for being here, everybody. During this lecture, of course, I'm not going to be getting into the mechanisms of cancer progression, nor am I going to get into cancer treatment, but we're going to be speaking about telomeres, aging, and disease. And there are some things that we can do right now, not only to slow the aging process, but extend maximum lifespan as well. So let's begin. I just want to make sure that my watch is set appropriately, because there's a lot of information here. We're going to start talking about telomeres, and just to back up again, you'll see that the telomeres are the caps on the end of the DNA. Telomerase is an enzyme that puts that cap back. What I want to start doing, because there is a lot of information, is I'm going to present a synopsis of my lecture. So anybody who wants to leave 5 minutes from now, at least you got the synopsis. So cells have a limited capacity to divide. And when a few telomeres become critically short, the cells enter a new phase called cellular replicative senescence. In a prospective population-based study, significant inverse correlations between baseline leukocyte telomere length and both cancer incidence and mortality were demonstrated, which emerged as independent of standard cancer risk factors. So what we're saying is shorter telomeres, increased incidence of cancer, increased mortality. Individuals with shorter leukocyte telomere length present a higher prevalence of cardiovascular risk factors and coronary artery lesions, a higher risk of cardiovascular mortality, and in particular of an acute myocardial infarction. Elevated markers of oxidative stress have been associated with shorter leukocyte telomere length. Telomere length has been shown to be significantly inversely correlated with lifetime depression duration. Leukocyte telomere shortening causes immunosenescence and impaired leukocyte function, which leads to increased inflammatory cytokine output, which then leads to increased oxidative stress, thereby promoting further leukocyte telomere length shortening. So you can see this becomes a vicious cycle. So basically, aging promotes aging. Interestingly, exercise has been shown to buffer the effects of chronic stress on leukocyte telomere shortening. Lymphocyte telomere length at age 60 was shown to be predictive of subsequent lifespan, with telomere lengths in the shortest quartile at age 60 having a 7 to 8-fold greater risk for dying of infection. By the transitive property, if elevated oxidative stress correlates with leukocyte telomere shortening, and leukocyte telomere shortening correlates with age-related disease and mortality, then elevated oxidative stress correlates with age-related disease and mortality. So that's one of the things that we're going to talk about, is to decrease the rate of telomeric degradation. You've got to get a handle, of course, on oxidative stress. Slowing the aging process and extending lifespan by decreasing rate of telomere degradation. The ways to do this are minimizing excessive oxidative stress. I have a whole lecture that we get into calorie restriction. There are different drugs that may actually extend lifespan. And one of the exciting ways to do that is with telomerase activation. And there are natural substances that are telomerase activators. And probably eventually we'll be coming out with drugs. And as we'll also mention, genetic engineering can be a very exciting modality for extending telomere length and hopefully extending maximum lifespan. So that's the synopsis. For anybody who wants to leave, you can, but please don't. So we'll get into telomere biology and carcinogenesis now. And probably most of you are familiar with the Hayflick limit. In 1961, Leonard Hayflick and his colleague Paul Moorhead discovered that cultured normal human cells have a limited capacity to divide, after which they stop and they enter a new phase called cellular replicative senescence. And these guys performed experiments that demonstrated that cells had a molecular counting mechanism. And once this clock is expired, cells are done dividing. The withdrawal from the cell cycle and into this new cellular replicative senescence phase is triggered as a result of shortened telomeres. Telomeres consist of many kilobases of TTA, GGG nucleotide repeats, and they are associated with a protein complex called shelterin. The telomeric TTA, GGG repeats shorten with each cell division, as well as from oxidative damage and other end processing events. So the two probably predominant factors that accelerate the shortening of the telomere is cell division as well as oxidative stress. When a few telomeres become critically shortened, there's a growth arrest state, at which time DNA damage signaling and cellular senescence is triggered. And cells can stay in this quiescent state for a long time. And as we'll discuss, this replicative senescent state may actually be protective against cancer. Now, human tumor cells derived from carcinomas almost universally bypass the cellular replicative senescence. And how do they do that? They do that by activating telomerase. So right now you should be thinking, interesting, telomerase can be a bad thing if it is allowing these cells that bypass cellular replicative senescence, it can allow them to survive and become a cancer cell. On the other hand, before you have cancer, if you express enough telomerase, you can prevent the critical shortening of the telomeres so you don't get cancer and you slow the aging process. And this is a pictorial. You'll see the green line or the green arrow at the top representing germline or embryonic stem cells, and their telomere length stays constant because of significant constant expression of telomerase. The pluripotent stem cells, they do secrete telomerase, but not to the same extent as the germline cells. And then the somatic or normal cells in red. Now, they do not significantly express telomerase. And as they continue to age, meaning there's continuous cell division, eventually their telomeres will become critically short. And they will hit what's called M1, which is a trigger for cellular replicative senescence, because the cells or the telomeres are simply too short to protect the ends of the DNA. Now, for the few cells that bypass that signal for cellular replicative senescence, they can keep dividing, but then they hit another phase that we call crisis, or M2, and it is the telomere is so short that the genome is incredibly unstable. And the rare cell that survives that does so by expressing telomerase and can become a cancer cell. So cells that have escaped crisis generally have two defining hallmarks: telomere stability, generally at short telomere lengths, and reactivation of telomerase. This suggests that the cancer stem cell was likely to have very short telomeres when telomerase was reactivated. Studies have shown that cancer cells with stem-like markers have similar or shorter telomeres compared to the bulk of the tumor. The vast majority of human cancer cells have telomeres generally the same or shorter than adjacent normal tissue, providing evidence that the cancer cell only expresses enough telomerase to protect itself from DNA damage. There's no advantage to making its telomere long, just long enough to sustain viability. So re-expression or upregulation of telomerase in cancer may reduce the ongoing chromosomal instability so these cancer cells can stay viable. In summary, telomere shortening may be a common underlying cause of chromosomal rearrangements in cancer. The function of normal tissue stem cells appears to change with increased age, and this may be due in part to telomere shortening. Tissue stem cells also show progressive shortening of telomeres with increased age, while the embryonic stem cells appear to fully maintain telomeres. And once again, this is due to full expression of telomerase. Similar to the embryonic stem cells, cancer cells maintain stable telomere lengths, whereas the normal tissue stem cells, as we showed in that prior pictorial, are mostly do not express telomerase, although they're telomerase competent. It is now widely accepted that replicative aging serves as a brake against malignancy. Cancer cells require multiple mutations to become malignant. Each mutation probably uses 20 to 40 divisions before achieving a population size sufficient for another spontaneous mutation to occur. So pre-malignant cells usually face the barrier of replicative senescence before accumulating enough mutated pathways to become frank malignancies. Once again, one critical step in oncogenesis involves the upregulation or reactivation of telomerase, and approximately 85 to 90% of all tumor biopsies are telomerase positive. Now, here's a very interesting study in the town of Bruneck in Italy. Telomere length and risk of incident cancer and mortality. The aim of this study was to assess the association between leukocyte telomere length and risk of both cancer manifestation and cancer mortality in this prospective population-based Bruneck study with near complete participation and follow-up and detailed information on all types of new-onset cancer available. So this is a population-based study with 1,000 individuals aged 40 to 79 who are randomly selected from the inhabitants of the town of Bruneck and examined every 5 years starting from 1990. The 1995 evaluation served as the baseline for the study. Telomere length measurements were performed in quadruplicate for the 1995 and 2005 samples, with the same individual samples positioned directly next to each other. During the 10-year follow-up, 92 of 787 participants developed cancer, incidence rate of 13.3 per 1,000 person years, in 44 of whom it was fatal. Compared with participants who remained free of cancer, the cases were older, physically less active, and more likely to be current or ex-smokers. Moreover, they consumed more alcohol and had lower levels of LDL cholesterol and higher levels of C-reactive protein. I have another lecture, and this is not the appropriate time for this, but I do want everybody out there to be cognizant of the correlation between low LDL and increased incidence for cancer. This was demonstrated with the Framingham data back in the 70s and is still present. There is clearly a correlation between low LDL and cancer incidence. So please research that when you get a chance. Very important. Baseline telomere length was substantially shorter in the participants with incident cancer than in those who remained free of cancer. A significant decrease in telomere length, of course, was seen with age. Men had shorter telomeres than women, and further associations were observed for the variables of diabetes, physical activity, vitamin D, presence of chronic infection, of course the inflammatory marker interleukin-6, and high levels of C-reactive protein. Telomere attrition was most pronounced in participants with the longest telomere length, whereas interestingly, and we'll show a slide, there was a paradoxical increase in telomere length in those with the shortest baseline telomere length. Short telomere length at baseline emerged as a significant and independent predictor of new onset cancer. The hazard ratio per 1 standard deviation decrease in telomere length for an incidence cancer was 1.6, which equals the excess risk attributable to a 12.8-year difference in chronological age. Compared with participants in the longest telomere length group, the multivariable hazard ratio for incidence cancer was 2.15 in the middle length group and 3.11 in the shortest length group. Incidence rates were 5.1 per 1,000 person years in the longest telomere length group, and it was 22.5 per 1,000 person years in the shortest length group. A total of 44 participants died of cancer. An association was seen between baseline telomere length and cancer mortality that surpassed the associations obtained for cardiovascular mortality and mortality from all other causes. Compared with participants with the longest telomere lengths, the multivariable hazard ratio for cancer mortality was 5.63 in the middle length group and 11.11 in the shortest length group. In absolute terms, 2, 14, and 28 of individuals in the longest, middle, and shortest telomere length groups respectively died of cancer. Furthermore, an analysis revealed telomere length to have a similar predictive value for cancer in both men and women and in various age groups. And so here is, once again, a pictorial. You'll see the shortest telomeres on top having, once again, the lowest cancer incidence, the shortest telomeres having the highest cancer incidence and the highest cancer mortality. And then on bottom, you'll see the longest telomeres very significantly having the lowest cancer incidence and lowest cancer mortality. So the comment from these authors, this is the first prospective population-based study to estimate the impact of telomere length on overall cancer manifestation and mortality. The authors demonstrated significant inverse correlations between baseline leukocyte telomere length and both cancer incidence and mortality, which emerged as independent of standard cancer risk factors. There was evidence of heterogeneous effects according to cancer type, whereas the tumors with a high fatality rate tend to exhibit more prominent relationships with telomere length, while tumors with a more favorable prognosis showed modest or no associations. I'm going to skip ahead a little bit to save some time. It is appealing to assume that the association between short leukocyte telomere length and cancer formation is partially mediated by the aging of the immune system itself. Aging of leukocytes reflected by short telomere length may impair immune surveillance and reduce the clearance of tumor cells. And in this pictorial here, you'll see at the bottom, very interestingly, with the shortest telomere length, you'll see the lines between 6,000 and 7,000 base pairs. Very interestingly, there was a slope up a little bit. So apparently, those with extremely short telomeres have an ability to express some telomerase. So it's tempting to speculate that cells with critically short telomere length may, under certain circumstances, reactivate the enzyme telomerase to escape from cell senescence and facilitate malignant transformation. Telomerase expression has been documented in normal peripheral blood leukocytes. T lymphocytes actually upregulate TERT on activation by immunoinflammatory stimuli, potentially linked to short telomere length. And TERT is the telomerase reverse transcriptase. I do want to point out there is an association also between circulating leukocyte telomere length and oxidative stress. In this study, they looked at leukocyte telomere length and cardiovascular disease. Individuals with shorter leukocyte telomere length present a higher prevalence of cardiovascular risk factors and coronary artery lesions, and a higher risk of cardiovascular mortality, and in particular acute MIs. This increased risk is independent of the classical risk factors like C-reactive protein, and strongly suggests a link between leukocyte telomere length shortening and coronary artery disease. Oxidative stress and inflammation are major factors that accelerate age-related telomere shortening, leading to the hypothesis that telomere attrition could serve as a biomarker of the cumulative burden of oxidative stress and inflammation. TTA-GGG is the repeating telomere DNA sequence or DNA base pair sequence due to the high guanine content telomeres are highly sensitive to reactive oxygen species. Emerging data suggests that the HMG-CoA reductase inhibitors, or the statins, have a beneficial effect on telomere biology, possibly contributing to the beneficial effects of statin therapy. Moreover, statin therapy has been shown to prevent telomere length erosion of the endothelial progenitor cells of healthy subjects, or after 12 months of intensive statin therapy in those with coronary artery disease. Okay, circulating leukocyte telomere length and oxidative stress. Statin therapy has demonstrated its efficacy in reducing coronary artery disease mortality in primary and secondary intervention trials, and it's been suggested that part of this beneficial effect, once again, could be its effect on telomere length. In this study, the authors found no associations between telomere length and demographic characteristics, treatments, or traditional risk factors, including LDL cholesterol, white cell count, and C-reactive protein. What they did find are new major determinants of circulating leukocyte telomere length, and these are markers of oxidative stress, FOS and OGG1. So 8-oxoguanine DNA glycosylase, or OGG1, is involved in a base excision repair pathway that has recently been shown to be active after telomeric DNA oxidative damage, and Finkel-Biscus-Jenkins osteosarcoma, or FOS, an ROS-induced transcription factor, is specifically involved in atherosclerosis-associated inflammation. FOS and OGG1 have recently come to light as new biomarkers of oxidative damage, as well as inflammation processes linked to telomere shortening. These markers, unfortunately, are not typically commercially available. Previous studies in humans have demonstrated the involvement of reactive oxygen species and inflammation in leukocyte telomere length variations. Demisi et al. Reported that systemic oxidative stress, assessed by urinary 8-epiprostaglandin F2, was associated with reduced leukocyte telomere length in hypertensive subjects. In a random population sample of 216 subjects, a higher level of circulating oxidized LDL was associated with shorter telomeres and, to a lesser degree, with increased stiffness of the carotid artery as an index of vascular aging. Moreover, in a large population of middle-aged men and women free of overt cardiovascular disease, telomere length was shorter in subjects with the inflammatory marker interleukin-6. This is an interesting study. Leukocyte telomere length in major depression, correlations with chronicity, inflammation, and oxidative stress, preliminary findings. Leukocyte telomere length was compared between 18 unmedicated major depressive disorder subjects and 17 controls, and was correlated with lifetime depression chronicity and peripheral markers of oxidation. And they used F2 isoprostane and to the vitamin C ratio, as well as interleukin-6. The authors examined telomere length within the depressed group as a function of cumulative lifetime duration of depression. Next, as a secondary test of their hypothesis, they dichotomized lifetime exposure to depression into a categorical independent variable, comparing the control subjects to the depressed subjects in the highest half of lifetime depression. Telomere length within the entire depressed group was significantly inversely correlated with lifetime depression duration. Individuals with greater lifetime duration of major depression had significantly shorter telomeres. When depressed individuals in the upper half of lifetime exposure to depression were compared to controls, significant differences in telomere length were observed. I'm going to skip ahead here a little bit. So, the bottom line is aging, as I talked about, promotes aging. Leukocyte telomere length shortening results in immunosenescence, or impaired immune function, impaired leukocyte function, leading to increased inflammatory cytokine output, to increase oxidative stress, thereby promoting further leukocyte telomere length, forming a vicious cycle. Now, interestingly, exercise. Some of you out there may be familiar with the mouse study. When they took mice and put them on the treadmill daily, they extended the maximum lifespan of the mice by approximately 20%. Well, it appears that exercise can buffer the effect of chronic stress on the telomere length. In the first study to link psychological stress with short telomere length, it was demonstrated that among premenopausal women caregiving for a chronically ill child, duration of caregiving, of course, was associated with telomere length. Yet, not everybody who is under this chronic stress experiences that kind of telomere shortening. So the authors proposed that physical activity can moderate the impact of stress on cell aging. Physical activity is related to longer telomeres, both in a national sample of twins and in endurance-trained athletes compared to sedentary controls. So the authors hypothesized that stressed yet physically active adults will be more protected from the impact of stress on their telomere length compared to those stressed and not active. Consistent with previous studies, the data in this study indicated that higher perceived psychological stress was associated with shorter telomere length. Importantly, however, physical activity was able to protect those experiencing stress by buffering its relationship with telomere length. These findings support the physical benefits of activity and, interestingly, map well onto Centers for Disease Control recommendations for amount of physical activity required to sustain a healthy body, which is 75 minutes of vigorous exercise per week. Greater than 6 minutes is the way we define vigorous exercise. The mechanism by which stress impacts the body at the cellular level are unknown, but once again, one likely pathway is telomerase activity. Chronically stressed individuals have lower telomerase activity and often demonstrate altered glucocorticoid profiles. Higher excretion of urinary stress hormones is related to lower telomerase and shorter telomeres. A recent experimental study demonstrated that exposing human lymphocytes in vitro to high levels of cortisol during primary stimulation and upon subsequent re-stimulation can dampen telomerase activity. Autonomic nervous system profiles associated with chronic stress, like high blood pressure, low heart rate variability, are also associated with low telomerase activity. Okay, so the data here indicated that above around 40 minutes of vigorous activity over 3 days, stress is no longer associated with short telomeres. 40 minutes over 3 days corresponds well to the CDC recommended levels of vigorous activity. So someone like me who is under a whole lot of stress, and I'm sure many of you are out there, exercise is extremely important. Recent longitudinal studies further underscore the notion that telomere shortening may constitute a biomarker for deleterious health status. In one study, lymphocyte telomere length at age 60 was shown to be predictive of subsequent lifespan. As we talked in the synopsis early on, the T-cell involvement in these associations is underscored by the fact that the individuals with telomere lengths in the shortest quartile at age 60 had a 7 to 8-fold greater risk of dying of infection as compared with those in the longest telomere quartile group. The Swedish so-called octo and nono longitudinal studies on elderly cohorts examined a variety of physiologic parameters associated with lifespan and identified a cluster of immune parameters that was strongly predictive of early mortality, independent of the cause of death. The so-called immune risk phenotype consisted of reversal of the normal ratio between the helper and suppressor T cells, reduced T cell proliferative potential, increased proportion of the T cells lacking CD28, indicating that they're senescent T cells, and evidence of CMV infection. So, summary of the conclusions so far. Critically short telomeres increase the risks of cells becoming malignant. Baseline leukocyte telomere length was an independent risk factor for both cancer incidence and mortality. Shorter leukocyte telomere length was associated with an increased prevalence of coronary artery lesions and increased risk of cardiovascular mortality. Lymphocyte telomere length at age 60 was shown to be predictive of subsequent lifespan. Oxidative stress and inflammation are major factors that accelerate age-related telomere shortening. Elevated oxidative stress markers are associated with shorter leukocyte telomere length, and so once again, by the transitive property. If elevated oxidative stress correlates with leukocyte telomere shortening, and leukocyte telomere shortening correlates with age-related disease and mortality, Then, by the transitive property, elevated oxidative stress may correlate with age-related disease and mortality. And if increased oxidative stress correlates with age-related disease and mortality, can nullifying this excessive oxidative stress affect the aging process as well as mortality? So if we change our lifestyle through calorie restriction, eliminating other habits that increase oxidative stress, participate in moderate exercise on a regular basis, consume potential telomerase activators, and ensure adequate intake of antioxidants, can we slow the degradation of our telomeres and improve the quality of our life while extending lifespan? Well, I do want to pause for a second and tell many of you that in the science world, the oxidative stress theory of aging is starting to fall a little bit to the wayside. And many of us, including me in my former years, take massive amounts of antioxidants. But what I do want you to understand is that oxidative stress is important for cell signaling. And very interestingly, in a mouse study with calorie restriction, the calorie-restricted mice have lived 20 to 25% longer. But when you take those same mice and calorie restrict them, and you give them a certain amount of antioxidants, they don't get the benefits anymore. And it seems that a certain amount of oxidative stress is necessary to stimulate the mitochondria to secrete more endogenous antioxidants. And what we found, or what the authors found, is when they gave enough antioxidants, they turned off the mitochondria's ability to generate endogenous antioxidants. So, and one of the interesting findings is that calorie restriction causes a chronic low level of oxidative stress. So there's a U-shaped curve on probably every single substance known to mankind. And the extremes don't appear to be beneficial. So I'm going to skip through these. These are just minerals, supplements, vitamins that have all been shown to correlate with longer telomere length. Now, you'll see vitamin D, magnesium, B12, but many of us assume vitamin C and E, that more is always better. And once again, I want you to keep in mind that there is a U-shaped curve and more may not necessarily be better. Okay. This made Nature news. The first documented reversal in a mammal. Telomerase reverses the aging process. Harvard researchers show first age reversal in a mammal. Telomerase activation was used to change old mice back to young adults. Brain, spleen, and reproductive organs were all rejuvenated. This resulted in increased neurons and new viable sperm cells. Sense of smell returned. Mice engineered to lack telomerase become prematurely decrepit, but they bounced back to health when the enzyme was replaced. The finding, published online in Nature, hints that some disorders characterized by early aging could be treated by boosting telomerase activity. It also offers the possibility that normal human aging could be slowed by reawakening the enzyme in cells where it has stopped working. This has implications for thinking about telomerase as a serious anti-aging drug or modality. When mice are engineered to lack telomerase completely, their telomeres progressively shorten over several generations. These animals age much faster than normal mice. They're barely fertile and suffer from age-related conditions such as osteoporosis, diabetes, and neurodegeneration, and of course they die young. DePinho's team engineered mice such that the inactivated telomerase could be switched back on by feeding the mice a chemical called 4-OHT. The researchers allowed the mice to grow to adulthood without the enzyme, and then they reactivated it for a month, and they reassessed the mice. What really caught us by surprise was the dramatic reversal of the effects we saw in these animals, a near Ponce de Leon effect. Shriveled testes grew back to normal, and the animals regained their fertility. Other organs, such as the spleen, liver, and intestines, recuperated from their degenerated state. The 1-month pulse of telomerase also reversed effects of aging in the brain. Mice with restored telomerase activity had noticeably larger brains than animals still lacking the enzyme, and neural progenitor cells, which produce new neurons, and supporting brain cells started working again. It gives us a sense that there's a point of return for age-associated disorders. Drugs that ramp up telomerase activity are worth pursuing as a potential treatment for rare disorders characterized by premature aging, and perhaps even for more common age-related conditions like aging. Gene therapy. Researchers at the Spanish National Cancer Research Center, led by its director Maria Blasco, have proved that mouse lifespan can be extended by the application in adult life of a single treatment acting directly on the animal's genes. The team treated adult 1-year-old and aged or 2-year-old mice with the gene therapy, delivering a rejuvenating effect in both cases, according to the authors. The mice treated at the age of 1 lived longer by 24%. Those treated at age 2 by 13%. The therapy furthermore produced an appreciable improvement in the animal's health, delaying the onset of age-related diseases like osteoporosis and insulin resistance, and achieving improved readings on aging indicators like neuromuscular coordination. The gene therapy utilized consisted of treating the animals with a DNA-modified virus, the viral genes replaced by the telomerase enzyme. This study shows that it's possible to develop a telomerase-based anti-aging gene therapy without increasing the incidence of cancer. The mice subjected to the gene therapy now under test are also free of cancer. Researchers believe this is because the therapy begins when the animals are adults, so they don't have time to develop the sufficient number of mutations to develop tumors. Okay, so this study is viewed primarily as proof of principle that telomerase gene therapy is a feasible and generally safe approach to improve health span and treat disorders associated with short telomeres. Now, this is a pictorial showing that with increased cell division on the x-axis, telomere length decreases. If it gets short enough and they express telomerase, once it's already got into crisis state, there's the possibility of the cancer cell. But if we can express telomerase either by taking a substance that is a telomerase activator or through genetic therapy, we can certainly increase the telomere length well before those telomeres reach the critically short state. And one of the substances out there is a single molecule isolated from astragalus, which has been shown to be a telomerase activator. And this is a pictorial showing that the percentage of critically short telomeres on the y-axis were significantly less after 12 months on a telomerase activator. And once again, mice treated with a telomerase activator, you'll see with on the red square is significantly red, indicating a lot of fatty liver, and then after being treated with the telomerase activator, certainly less fat accumulation. On the bottom slides, you'll see the control and the telomerase activator where they had skin and hair resected, and then after 2 weeks of receiving a telomerase activator, you can see the nice growth. So, very quickly, a new paradigm for life extension. Measure baseline telomere status. Measure the percentage of critically short telomeres. Measure the median telomere length. Measure indicators of oxidative stress like interleukin-6, F2 isoprostane, CRP, but keep in mind that C-reactive protein is a very, very labile protein. Measure indications of immune status like the CD4 to CD8 ratio. Measure the mature T cells, CD3 and natural killer cells. Measure antioxidant status, vitamins and minerals. And then lifestyle changes as indicated. Begin an antioxidant regimen only as needed, not to get supraphysiologic antioxidant levels. Consider other natural telomerase activators. And after you've been on this regimen, after a few months, remeasure oxidative stress indicators. And then 6 to 12 months later, repeat telomere measurements. I want to finish up by saying that there are exciting techniques on the way, both from a drug standpoint as well as from gene therapy, that I believe in our lifetime we will be able to significantly extend lifespan. Thank you very much.